There are three terms to describe this certain infectious disease from history, and they are the White Plague, Tabes, and consumption. Do you know what these three terms might characterize? What infectious disease? Also, King Tutankhamun of ancient Egypt, as well as Eleanor Roosevelt and Ho Chi Minh died of this disease. Do you know what this disease is? If you don't know the answer is tuberculosis. So my name is Chloe Ilagan, and I will be talking to you about tuberculosis, TB. So tuberculosis is an infectious bacterial disease that attacks prominently the lungs. It can also attack other body parts, but it does mainly attack the lungs. There are two terms, pulmonary TB, which refers to TB in the lungs, and then extrapulmonary, which refers to other body parts. And these include the kidneys, the spine, etc. And there are two related conditions that relate to this disease. LTBI stands for latent TB infection, which refers to the TB bacteria residing in our body, but not producing any signs or symptoms. So it's just there, and the person doesn't get sick. There's also TB disease, which is the actual multiplication and replication of TB bacteria within the body, and that person experiences symptoms, and it's transmitted through airdrop nuclei. So when a person inhales TB bacteria. The causative agent of this infectious disease is Mycobacterium tuberculosis, and the shape is bacillus, so it has that rod shape to it, and it's a strict aerobe, meaning that it grows and survives in environments that have the presence of oxygen. An interesting characteristic also about this bacterium is that it has a waxy cell wall due to the presence of mycolic acid, so it's acid fast. So this kind of relates to the stain that we did in microbiology lab. Acid fast meaning that it stains orange or like a bright red color because of that carbofuchsin primary stain. So mycolic acid resists decolorization. So once that carbofuchsin is stained and then the decolorizing agent of acid alcohol is added, it doesn't wash away. So it just keeps that orange stain and doesn't stain blue, which non-acid fast bacteria do. And it's most recognizable in that Zeal-Nielsen stain, so it's a hot method of the acid stain, not the cold method. It's a pathogen of mammalian respiratory system, and the human host is going to be the natural reservoir for this bacterium. So this is just a depiction of it, the waxy cell wall, and as you can see, the rod shaped, as well as the bright red stain from this acid stain, and it needs oxygen. The pathology of this bacterium is quite simple. So for example, a human, it'll inhale the TB bacteria, and it's the droplet nuclei that enters the lungs. That droplet nuclei contains tubercle bacilli and goes all the way deep inside the lungs to the alveoli tissue. And then the tubercle bacilli will continue to multiply, and then it goes to the bloodstream and then to the rest of the body. We also have the macrophages in the next step that ingests the tubercle bacilli. The macrophages are specialized immune systems that surround it, and then it forms a granuloma. Granuloma is going to refer to that shell-like protective barrier around the tubercle bacilli. As you can see, these gray cells. And so once a granuloma forms, it kind of keeps the tubercle bacilli contained and controlled. That's when we have LTBI, which is that latent TB infection. So there's no bacilli exiting or spreading throughout the body. But if the granuloma breaks, on the other hand, we have TB disease, because obviously the tubercle bacilli is going to go out, spread to the rest of the body, and then the person's going to experience symptoms. And keep in mind that this does not have to happen in the lungs only. It can occur in any part of the body. The history regarding this disease is quite interesting. We're really familiar with Robert Koch, but he was the one that discovered and identified Mycobacterium tuberculosis on March 24th, 1882. Then he later, on April 10th, 1882, published the Etiology of Tuberculosis, which just goes into detail describing and characterizing the causative agent, this bacterium, to produce tuberculosis. And then Koch's postulates, he wrote in 1890, which is the main paper that serves as the fundamental basis for the causes of infectious diseases. And that is how we're able to see if whether or not a microorganism is the certain cause for an infectious disease. Historical developments that also did occur that were very, very pertinent to this disease includes Paul Ehrlich's discovery of the acid-fast nature. So we see how the bacterium functions due to that resisting of the decolorizing agent acid alcohol. And this is Paul Ehrlich right here. Von Perquet and Manteau, they developed the tuberculin skin test in 1907 to 1908. This tuberculin refers to the sterile extract protein that is obtained from the culture tubercle bacilli, and it involves in the diagnosis of tuberculosis. And then Florence Sieber, he prepared the purified protein derivative of tuberculin. It's also called PPD, and this happened in 1931. So this skin test is going to be right here. A positive test is to see whether or not you have tuberculosis. And keep in mind, people at high risk with tuberculosis are the ones that usually get this skin test. So if you are not really at risk, then they really do suggest not to get this test. But a patient is going to be intradermally injected with this tuberculin, and then after 48 to 72 hours, the doctor is going to check on you to see whether or not there's redness or swelling. So that redness or swelling is going to be the sign of a positive test for tuberculosis. And then obviously negative test is going to be no redness, no swelling, nothing happened. And it's important to know that tuberculosis is not common in the US. It's more common in third world countries, kind of in like Asia and Africa. And so they really do not perform these skin tests in the US. So that brings me to the prevalence in non-US countries. So we have China, India, Bangladesh, Nigeria, Pakistan, South Africa, Indonesia, and the Philippines as the 8 main countries that make up 2/3 of the new total TB cases. This was recently in 2020 when 86% of new TB cases occurred in the 30 high TB burden cases. So 8 countries of these 30 have a lot of people with tuberculosis infection. And it kind of started off in the 18th century when in Western Europe there were a lot of epidemics that had tuberculosis, and plus this part of the world, third world country, kind of harsh and severe conditions, different living conditions in the US. So we see how the prevalence kind of makes sense that it's going to be here. The most interesting thing that I found when researching tuberculosis was how scientists were able to differentiate between pathogenicities between these strains of tuberculosis. So there's Erdmann, H37RV, and CDC 1551, with CDC 1551 being the least virulent, and then Erdmann being the most virulent. So these pathogenic strains are very, very important to look at. And so in 2003, scientists used the New Zealand white rabbit model. And so these scientists, they bought and purchased these white rabbits from New Zealand and made sure that they were maintained healthily so that they were ready for this experiment. They were healthy for this. And so just prior to infection, the volume of air that each rabbit breathed per minute was calculated from the respiratory rate, the tidal volume of air inhaled by each rabbit, as well as the per minute ventilation. The aerosol was then generated with a colocin nebulizer containing that bacilli, and the aperture of the nebulizer disperses these clumps of bacilli into units of 1 to 3 bacilli. Throughout the 10-minute exposure period, a small sampling port was placed in the exposure chamber box at the level of the animal's nose, and it was used to collect the samples. So for each rabbit, the number of viable bacilli inhaled was calculated based on the volume of inhaled air during exposure and the number of CFU, colony forming units, cultured from the samples per milliliter. These rabbits were housed in biosafety level 3 facilities at the George Washington University Medical Center immediately following infection. And then 4 weeks after infection, a dilution of tuberculin was injected intradermally. And then 2 days later, the resulting skin reaction was measured to see that redness and swelling. And then at 5 weeks after infection, the rabbits were euthanized with pentobarbital intravenously, and the number of visible primary tubercles in the lungs was counted. So we see that Erdmann was the most virulent because it required the least amount of inhaled bacilli to form one tubercle, and the tubercle is going to be like a lesion. So it only required 320 to 827 bacilli, as shown here in this graph. And Erdmann strain is going to be the solid line. And then for H37RV, 586 to 1889 bacilli were required. So a little bit more, so a little bit less virulent, but it's going to be shown in this dotted line here. And then CDC 1551 is the least virulent with 1667 bacilli needed in order to form just one tubercle. So we see the difference in virulence, and it's really, really interesting. And this New Zealand white rabbit model was not used for the first time in 2003, but it was also used in 1898 by Theobald Smith. And he used this to determine the difference in pathogenicity between M. tuberculosis and M. bovis. M. bovis is a different species in the family of Mycobacterium that is quite similar, but not exactly the same as tuberculosis. So he noticed that tuberculosis, when it was injected into the rabbits of 10, 1 out of 10 rabbits died. But for M. bovis, of a similar dose, 9 out of 9 rabbits died. And so all of these rabbits died, meaning that it was way more pathogenic than M. tuberculosis. And so we have a picture here of the rabbits. But that is mainly it for tuberculosis, and I wanted to thank you for watching. These are my references and then my picture references. Thank you so much.